I'm Myra Sneem. I'm doing a PhD in microbiology and molecular genetics from University of the Punjab. The research study which I'm going to present today is Kupchi Desert, China, a promising source of antimicrobial and in vitro anti-cancer echinobacteria. In this presentation, I will talk about how the study was important, why it was needed, and what purpose it would serve. What role echinobacteria play in drug discovery, the introduction to Kupchi Desert, China, what are the objectives of the study conducted, the methodology followed, results, and what was concluded from it. If we talk about the importance, there are many factors which have compelled the scientists to discover novel bioactive drugs from natural resources, and one of the major factors is drug resistance in pathogens, both multi-drug resistance and extensive drug resistance. This causes the pathogens to escape the drug action pathways, and there are many evasion mechanisms which they follow. One of the major causes of drug resistance is extensive use of antibiotics or using antibiotics without proper prescription. Other factor is prevalence of chronic cancer cases worldwide. According to WHO reports of the year 2020, cancer is the second leading cause of death globally, which estimated 9.6 million deaths in 2018. So, finding effective anti-cancer drugs is the need of the hour. The third factor is the inefficiency of available cancer chemotherapeutics. The available treatments are either toxic or show undesirable side effects. All these and many other factors alarming the situation worldwide, and more research is to be done on natural resources of bioactive compound producers. Actinobacteria are soil-living gram-positive bacteria having high guanine and cytosine content in their DNA, making them more stable to denaturation. They are distributed both in aquatic and terrestrial ecosystems. Actinobacteria have played a significant role in human health in the last decade throughout the world. They are considered to be the most prolific producers of secondary metabolites. They have extensive secondary metabolism and produce a large number of drugs which are currently in use, including anti-cancer, anti-allergic, and antifungal compounds. Cyanobacteria play key roles in terrestrial ecosystem functioning, like they are very promising producers of bioactive compounds. They contribute to global carbon cycling and a major content of warm, dry soils, like up to 62% consists of cyanobacteria, and they also help in increasing the plant productivity. Streptomyces is the largest genus of Actinobacteria with over 500 reported species, and more than 80% of antibiotics are produced by Streptomyces. They produce spores and are noted for their distinct earthy smell, which results from the production of a volatile metabolite geosmin. Among 45% of the presently known bioactive microbial metabolites, over 10,000 compounds were isolated from various Actinomycete species, 34% from Streptomyces, and 11% from the rare Actinomyces. Streptomyces are the most frequent producers of antibiotics compound, with approximately 77,600 compounds produced by Streptomyces, while rare actinomyces produce 2,500 compounds. Researchers are more interested in exploring untapped habitats for novel drug discovery. That is the reason we chose Kupchi Desert, China, located in Inner Mongolia, to isolate the actinobacteria for getting novel drugs. Deserts are extreme environments that are characterized by extreme aridity, intense solar UV radiation, and extreme shifts of temperature in day and night cycles. The Ordos Desert is a steppe region lying on plateau in the south of the Inner Mongolian Autonomous Region of the People's Republic of China. The area of Ordus Desert is 90,650 kilometers square. The soil of the Ordus is a mixture of clay and sand, and that is the reason that it's poorly suitable for agriculture. The Ordus Desert comprises two large deserts. One is the Kupchi Desert, which is the seventh largest desert in China and located in the north of Ordus Desert. And the second is the Morse Desert, which is eighth largest desert in China. Our sampling source was from Kupchi Desert, China, which is in Inner Mongolia, and its area is 18,600 square kilometers. The diversity of the cyanobacteria in this habitat and the bioactive compounds that they produce have been poorly explored. So this would be the first study of its kind to explore Kupchi Desert, China for actinobacterial isolation and compound purification. No such study has been reported yet. The objectives of this study included isolation and purification of strains, strain characterization through polyphasic chemotaxonomic approach, preliminary biological and chemical screening, purification and structural elucidation of bioactive compounds. The major steps which were followed in this research was the isolation and purification of the Cyanobacteria strains through some specific media and their morphological, biochemical, physiological, and genetic analysis, preliminary biological and chemical screening, and lastly, the purification and structural elucidation of bioactive compounds. After the preliminary biological and chemical screening, 12 strains from the collection were selected as priority strains for scale-up fermentation studies and the purification and identification of active compounds which are produced by those strains. So, some of the strains showed presence of new derivatives of compounds, which showed good activity against pathogenic gram-positive as well as gram-negative bacteria and cancer cell lines. The study revealed that the cyanobacterial strains originated from this site are a promising source of clinically useful antimicrobials and various other chemotherapeutic agents. Screening of such potential actinobacterial strains would help to strengthen our capacity against the rapid proliferation of different tumor cells and against antibiotic resistance. At the same time, it can be helpful to solve several health problems regarding cancer and infectious diseases. Overall, this study may yield some commercially useful actinobacterial strains and compounds.